It's my pleasure to welcome Professor Jürgen Hahn. He's going to be talking to us today about the development of a diagnostic test for autism spectrum disorder where big data meets metabolomics. Professor Hahn is the department head of the Department of Biomedical Engineering at Rensselaer Polytechnic Institute and also holds an appointment in the Department of Chemical and Biological Engineering. A little bit of background on his education is he received his diploma degree in engineering from RWTH Aachen, Germany in 1997, and his master's and PhD degrees in chemical engineering from the University of Texas at Austin in 98 and 2002, respectively. He was a postdoctoral researcher for the chair of process systems engineering at RWTH Aachen, Germany, before joining the Department of Chemical Engineering at Texas A&M University in College Station in 2003. And then he moved in 2012 to Rensselaer Polytechnic Institute, where he is now. His research interests include systems biology and process modeling analysis. He has over 125 peer-reviewed publications in print. Dr. Hahn is a recipient of the Fulbright Scholarship in 95-96, and he's received several referee and editor appointments. And for example, he was a Best Referee Award in 2004 from the Journal of Process Control, CPC7 Outstanding Contributed Paper Award in 2006, and he's the Outstanding Reviewer by the Journal Automatica in 2005, 2006, 2007. And then in 2010, he was the Computing and Systems Technology Division of AICHE Outstanding Young Researcher. He's been elected as an AIMBE Fellow in 2013, and he served as the IEEE CSS Board of Governors in 2016. He's also been a CASH trustee since 2014. He's currently serving as the associate editor for the journals Control Engineering Practice, Processes, and Journal of Process Control. It's my pleasure to welcome today Professor Jürgen Hahn.
Okay, well, thank you, John, for the invitation as well as the introduction. And so, as you got from the title, I'll talk about how systems analysis techniques can be applied to metabolomic data in order to basically make some headway on diagnosing autism. And let me give you a little bit of background. Yeah, so autism spectrum disorder, as the name already implies, because the word spectrum is there, you find that this is actually really a large group of diseases, and the way they're characterized by is really just by two things that people are looking for. One are these difficulties with social communication interaction, and the other one are expression of restricted repetitive behaviors and interests. As you can see, these are really behavioral measures. This is not something where they give you any insight of what's happening in the body. The problem with that is that if you ever want to talk about how you would go about treating autism, you need to kind of know what's happening in the body. But if you can't even diagnose it other than by just observation, as is currently the case, this is a challenge. On top of that, autism presents a few other challenges. One thing is that usually the autism diagnosis goes together with a number of co-occurring conditions. I have a few listed here, but there's a huge number of children that have anything from intellectual disability, ADHD, epilepsy, or any type of GI problems, and so on, or even the combination of those, and they often go together with autism. So it is not just that you can really separate it as one aspect of the disease, and these are all separate things. At some level, they're interconnected. What makes autism, what makes it very timely to look at this whole field is that when you basically monitor the number of cases that you have with different conditions over time, you actually see that there is a significant increase in autism over time. One thing I want to notice here is that unfortunately, there is a lot of really, misleading information in the public, information on this. Part of it is like some people go back to the 1970s when there were very, very few cases of autism diagnosed, and say, well, this has been like an exponential increase, and this may just continue into the future. That's not particularly meaningful, in part because we changed diagnostic criteria, and quite frankly, the data from the 1970s was not collected with the care that we use nowadays with these things. At the same time, the reason I'm comfortable showing this graph here is that between about roughly the mid-1990s and around 2010, we actually did not change the diagnostic criteria, and the collection of data was at a similar level. And yet you still a significant increase, whereas most of these other conditions are somewhat flat. So it's certainly something that needs to be investigated, and that makes it very timely to look into this problem. A few other facts about autism, and this comes straight from the CDC website. So currently it affects roughly 1.5%, so 1 in 68 children. And for some reason that we don't really understand, autism is 4 to 5 times more common in boys than in girls. And you find that approximately 83% they have a core occurrence with one or more non-SD related conditions, some of the ones that I've talked about on the previous slide. Some people would argue that if you're looking at the more severe cases, this is probably closer to 100%, but it's not always easy to diagnose, because if you have somebody who's nonverbal, this could be difficult. Yeah. Looking at the economic burden for this, that really puts things in perspective. I mean, in 2015, the U.S. spent roughly $265 billion, that's billion with a B, to care for people with ASD. If you look at the individual, that the average currently over lifetime is 1.4 million, and approximately 2.5 million if there's intellectual disability. In fact, if you look at other diseases that get a lot of attention from depression, like depression, ADHD, Alzheimer's, or diabetes, you find the cost for autism actually exceeds those. It's certainly in the same realm. One of the reasons you tend to hear a little bit less about this is simply the cost oftentimes is different. Like in many of these conditions, it's directly medical costs that's relatively easy to, or a lot of it is medical costs that's relatively easy to track. But for autism, for example, we're talking about special education classes. These are not paid by the federal government. They're usually paid by your school districts. And so it's harder to get that information, but it's out there. It clearly is a very significant problem. And unfortunately, it's a problem where we know very little about. And this is kind of where my work comes in, to basically help push that forward a bit. So, as I mentioned earlier, there's a lot of controversies about many of the aspects with autism. From where does it come from? What could be done about it? But there's one or two aspects that everybody agrees on. And one is that the earlier you can diagnose, the earlier you can start some treatments, which will lead to better outcomes. I should add here, given that we don't really know the condition in the body that goes along with autism, these treatments are oftentimes not medical treatments. They're things like special education services. But everybody agrees the earlier the diagnosis, the earlier you can start treatments, the long-term outcomes will be better. Yeah. The challenge is that given that you don't have any lab test for autism, I mean, the diagnosis right now is what's called the clinical diagnosis. Basically, the parent brings the child to a developmental specialist, and they observe this child for an hour or two and determine how does the child interact with the parents, with toys, and then determines that based on this interaction, if the child has autism or if they're developing normally, but maybe they're just developing slower in some aspects, which is very, very common for children who are very young. Yeah, and so this is not easy to do. And the challenge with this is that, yes, while everybody agrees that if everybody's on top of it and you have a good team of doctors, you can diagnose as early as 18 to 24 months. Currently, the average autism diagnosis is done at 4 years. So meaning there's 2 to 2.5 years of time that are essentially lost because you can't start any treatment unless you have a diagnosis first. And so if you had a test that allows you to basically push the date of the diagnosis to a younger age, that would be really, really helpful. And, now, let me give you one more piece of information that I think is important to put things in a broader context. And there's something a little bit here about the history of what we know about autism. Like if you look at early twin studies, people thought that autism was almost exclusively genetic, and don't get fooled here. The paper by Gogler, the paper's from 2015, but it's a review paper. So these studies are from 15, 20 years ago. They're not very recent. If you look at more recent studies, however, it becomes more of a mix. I mean, there clearly is a strong genetic component in autism, and everybody agrees on that. But there are also environmental factors that come into place. What's unclear is, for example, if these environmental factors are separate from the genetic factors, or if the genes basically provide a predisposition that then, when environmental factors come into play, result in the condition worsening. And so if you basically look at the current state of the art, you find that really half of what we deem, half of the contribution to autism is likely genetic, and a little less than half is environmental factors. What's really important here is we spend almost $1 billion trying to find out what are genes that are underlying autism with very limited success. There's like these single-digit percentage cases where we really can attribute genes to autism. The rest, like this huge number here of the 49%, this ranges anywhere from dozens to hundreds of genes that may have very, very small implications, but pretty much almost nobody has all of them at the same time. So we cannot really diagnose anybody with autism just based upon the genes, unless they have one of these very rare mutations like Fragile X is one of them. And so what we wanted to do with them is say, look, I think you probably have to look at the genes and environmental factors together. And the best way to do that is that you're looking at certain different systems in the body. And many things that we know are prevalent in a lot of children that have autism. Like, for example, there's the gut-brain axis. We know the immune system, there's some component to it. What we decided to do here is, however, we just focused on two. One of them is oxidative stress, because that's been found to be very prevalent in children with autism. And the other one is epigenetics. And the reason I say that is that a lot of the genetic work, people start talking about that, maybe the epigenetics, essentially the process of controlling which genes get turned on and off, that may play a major factor and not just the genes itself. And so we said, fine, let's look at those two aspects, because oxidative stress can be somewhat tied to environmental factors. The epigenetics will affect the genetic factors that we're seeing. Yeah, and in order to do this, we look at what are pathways in mammalian cells that basically contribute to this. And there's the folate-1 carbon metabolism and the transsulfuration pathway. Here, what I'm kind of circling here, this is the folate-1 carbon metabolism, and the transsulfuration is this part down here. And what you find is that this, the folate metabolism, basically where SAM gets converted to SAW here. This regulates DNA methylation, and DNA methylation is the regulator for epigenetics. So this is where basically the link to epigenetics comes in. At the same time, the transsulfuration pathway down here leads to the production of glutathione, and that's your body's most powerful antioxidant. And related to that, I mean, oxidative stress is related to detoxification, if you hear these terms. And so, so basically, this is all tied together using these two pathways. So we decided, let's focus on these two particular pathways. But what we wanted to do in the first step is that rather than just run a completely new study from scratch, we decided, let's have a look at what's already out in the literature. And there was a very good study that was published a little over 5 years ago at the University of Arkansas for Medical Sciences. And in this kind of study, they focused on these two pathways. They had a study with roughly 160 participants, half of them on the autism spectrum, the other half were typically developing. And they also had as another group to look at the siblings, about 47 siblings of the children who had ASD, but these siblings are actually typically developing. So you would have to lump them together with this group, but keep in mind they share many of the genes that the people in the ASD group have. But we wanted to see where would those fall. We would expect them to fall somewhere in between, but since they don't have autism, likely closer to this group. But let's see what the results show. Furthermore, we had 24 measurements from this pathway. Yeah, and these are the metabolites listed, as well as things computed from the metabolites, like some ratios or percentage of DNA with methylation, oxidized glutathione. Last but not least, I mean, the autism diagnosis usually is just a diagnosis, either you have it or you don't, but There's, given that it's a spectrum disorder, some cases are more mild, while others are a lot more severe. And so you have a number of measures that try to basically measure certain aspects of ASD severity, and one of them is the Vineland Adaptive Behavior Composite Score. And what this is based upon is that a lot of children with autism struggle in their adaptive behavior to adapt to changes, new environments, and so they tend to score lower on this. And usually, the lower they score, the more severe the case. So we basically said, well, let's use the data from this study. I should say that when this study was published, they found that there are some statistical differences between the groups, but if they were to give you measurements from any single individual, you cannot tell which group this individual falls into. So for diagnostic purposes, this does not do the job. And the reason, one of the concerns that we had is that all the analysis was done what we call univariate analysis. Like they look at, for example, the measurements for methionine, what is the measurement, and what's the standard deviation, and basically what's the measurements for people in this group versus people in this group. And the groups overlap very heavily, so you can't do that. What we decided to do instead, we said, let's look at multivariable techniques, such as Fisher discriminant analysis. The idea here is that if I have two groups, I want to separate them as best as possible. Now, this is cursed for linear case. Let's assume here you have this group of basically blue measurements, and you have a group of red. And I'm going to focus just on two different measurements. I have X1 and X2, and each one of those would correspond to one individual. If I just wanted to classify this here based upon measurement at X1, you see that basically the red and the blue, they heavily overlap. If you just look at X1, there's no way you could tell if somebody belongs to the blue or the red group. Similarly, if you look at X2, you find that yes, on average, this population has a slightly higher average, but still there's a lot of overlap between in the measurements of X2 between the red and the blue group, and you cannot separate them. But clearly, if I were to draw a line here in between, and something basically project these points to a line perpendicular to what I've just drawn, this indicates that everything to the left on this line is red and everything to the blue is right. So if you use this multivariate information, we are able to separate this. And the key here is to not just restrict yourself to 2 dimensions, but we can take multiple measurements. Clearly, it becomes more difficult to show them. Now, if you have nonlinearities, you can also do this as well. Like we can use something like kernel Fisher discriminant analysis, and here we use Gaussian kernels. And there you can basically see that, well, if I project everything on the surface, then I can find, and I cut through the surface, everything above is basically blue, everything below is red. So I can separate this. So nonlinear behaviors are possible as well. I don't just have to cut through lines if I'm interested in this. Last point to consider here is, and this is really important, especially when we look at multivariate system. These are basically our patients. And what we want that when we try to develop a model that separates our patients in 2 or more groups, where 2 is what we're interested in here, then you want to make sure that the model basically reflects your patients really well. But the way you do this is that I collect data from the patients, and then I basically develop my model based upon the data that I have. And the idea is that, well, if my data represents the system well, and my model works well on the data, then hopefully my model should work well on the system. But that's not guaranteed, and then because you have this problem of overfitting. If your model works too well on the data, most likely your model will actually not work well on the system itself. Yeah, to just give a brief example here. So if I were to tell you this is made up, of course, yeah, but it makes the point. So if I take a true model, that's a polynomial here of 4th order, and it is shown in blue, and I take 5 measurements and I add some noise, and so these are here these pink dots. And if I were to then try to estimate a model, and I'm even cheating here for estimating, I'm using a model that has the exact same structure, which in reality you never even know. What you find is then, yes, my model fit exactly goes to the point, so your sum of squared error is 0, so would look like a perfect fit. But if I compare my black curve to the blue, this is clearly not a good fit at all. I mean, out here the predictions are horrible, but even within this range, the dynamics exhibited by the black line is very, very different. So why? Well, because we had 5 data points, we fitted 5 parameters. That's about as bad as it gets, but the reality is that we should always be careful about how many parameters we can fit. If you pick something simple like this red model, then you find that even though there's no mismatch between the real model and what I'm measuring, but the red one is much closer to the blue, the original model, than what we get here. And the key is, this is because of overfitting. So you're going to have to use some form of cross-validation. And what we decided to do is we use leaf-on-out cross-validation. So traditionally, you have your samples, you develop a model on the samples, and then you basically compute the errors that you get between your model predictions and what your samples are. What we want to do with cross-validation is we actually take one sample out. We develop a model upon the rest, and then the model only predicts the data, this is just for prediction, which was left out, and you get an error. I put this data back into my set, and I remove the next one. I develop another model and predict what the error is. And I go through this until each data point has been left out, and I have a number of errors here. And these are the ones that I then want to minimize. And that tells me how complex my model structure could be, because you really find if your model gets too complex, you are overfitting, and these errors will go actually up then. And so now once I have the right model structure, I can go back and use this model structure on the entire set to come up with the model that I want. So, and if I apply this to our data to our autism data, and you actually can find that here the red group, which is the neurotypical or typically developing participants, separate quite nicely from the children who are on the autism spectrum. So if I look at the probability density function, there's a little bit of overlap here, but you get more than 95% right that you can actually predict. So let's make this more difficult and say, so what happened with the siblings? Because, I mean, after all, the siblings should be genetically much closer to their brothers and sisters who are on the autism spectrum. At the same time, they don't have the condition. So we would expect that they're somewhere in between, but they should be closer to the neurotypical participants. And that's exactly where they turn out to be. Yeah, they are not. It's not a perfect overlap with the typically developing group, but it's much closer there than with the ASD group. Yes, here, if you were to do a diagnosis, your misdiagnosis becomes larger. In practice, this is a complete non-issue, because if you're diagnosing somebody, you're going to ask, do you have siblings who are on the spectrum already? So you can take this information into account. But this is kind of exactly what we expect it to be. So this is nice validation. And keep in mind, the siblings, they're just used for testing here. None of the information from them has been used at all for building the model. One thing that we want to stress here is that even though I have 160 or 159 participants, not counting the siblings, you want to use a subset of variables as inputs. And so we figure out what are the most important ones, and we basically did some, in this case, we used the brute force approach to really determine what are the key, what is the subset of measurements that gives me the best results. And you find that using this, the groups of total of 7 variables, they separate very nicely. And what's important here, things like DNA methylation, which directly is tied to epigenetics, this comes out to be one of the key variables. Similarly, here the percent oxidized glutathione, this is directly related to the oxidative stress. So the variables that turn out to be the most important ones are exactly tied to the reasons why we choose to focus on these pathways in the first place. So it's nice verification of that. So if we apply this data then to the original, or this classifier to the original data set, you find out of the 83 participants with autism, 81 of them we would classify as having the condition, 2 of them are misclassified. And out of the 76 who are neurotypical, we basically determined that yes, 73 of them indeed do not have autism, and there's 3 who would get a false diagnosis. So in summary, we get very high accuracy here. If you don't like the misclassification errors like the type 1 and type 2 errors that we get, for example, some people says, no, I don't want anybody who doesn't have the condition to ever get misdiagnosed. that's fine. All you have to do is modify the cutoffs. I mean, then your other type of error is going to get a little worse, but this is acceptable for what we have. We really just need 7 key inputs here. You don't have to measure all the ones that we did. And as already said, some of them are tied to oxidative stress and DNA methylation. So our assumption for looking at this was the right one. Yeah. So far here, basically, we focused on just the diagnosis, classification. But you want to know is that, could I actually tell something about the severity of the condition? And so we have to do regression. Yeah, and what we're interested in is having these metabolite measurements. Can I predict essentially the output measure, in this case, the Vineland score from these measurements? And keep in mind that we really have to watch out here, particularly if you use nonlinear model, it's very easy to overfit. And so that's, we again use cross-validation in order to handle that. So traditionally, if you use ordinary least squares, you have your input set and you have your output set, and you try to find out essentially what's the best mapping. If you use principal component in a regression, you also try to remove the unimportant correlations in X. If you basically use partial least squares, then you remove the unimportant correlations in X, as well as in the outputs Y, because they appear here again, as well as between those two. So this actually makes it robust even for relatively small data sets, and that's very, very common for these types of problems. If you need a nonlinear relationship, then you basically cut this open, send this through a nonlinear transformation, and you perform the same thing. This would get you to, for example, kernel partial least squares is one of the extensions. So if we did this here, turns out we need about 5 variables to get the maximum prediction accuracy. And if you look at the predicted versus the actual Vineland score, we get an R-squared of 0.45. Keep in mind, this is for medical data, this is fantastic. I mean, there are plenty of conditions that we treat based upon R-squared of 0.1 or 0.2. So 0.45 is very high. You cannot compare this to chemical plant data where we want R-squares that are way above 0.9. This just hasn't happened for these types of problems, but this is a very high R-squared for what we're going for. So in summary for this part, as of now, we don't have any real biomarkers we could be using, but using the results from our study here, we've shown that actually we can predict if somebody would have has ASD or if they're typically developing. And this is with a very high degree of accuracy. So there's the impact of this can be tremendous. Yeah, and we can also basically relate some of the measures of ASD symptoms from, we can correlate that with the metabolite measurements. So the potential is very significant. What's key here, however, is that like in any medical intervention or medical investigation, you first have to replicate this on another study. And so not surprisingly, we got a lot of coverage for this. I mean, it made the journal cover. That was not unexpected, and it only took a few days, and people actually wrote papers about our paper already. And this was really then covered everywhere from Thomson Reuters, Netscape, and pretty much you name it. I mean, there were over 200 newspapers and TV stations that actually picked up on this. So it's the impact or the potential impact can really not be overstated. Let me show some hot off the press results here. These are unpublished as of now. So as I said, we need to replicate these results in a new study, but starting a new clinical trial is costly. So what we first did, saying, well, is there another study that we can use? Unfortunately, there's no study that has all the information, but I have a study that has only children on the spectrum, but nobody who's typically developing, or at least I don't have the data measurements for it. Unfortunately, they're not measuring our 24 metabolites, so I left 2 of them out. And so if I leave those 2 out, then basically we have to develop a new model without those 2 measurements, see how well this works. This is down here. And then we basically convert a computer or PDFs for the original cohort with autism and the typically developing ones. And here would be a cutoff. And now we apply the new data set to this, and you find that this new data set with 156 individuals also with ASD, it nicely matches the previous one. It's not a perfect match in the sense that the tail here is much shorter than this, But that's not unexpected if you're talking about studies that were taken years apart, and even the measurements are done by different groups, and the protocols vary a little bit. So, but this is very promising, and we are currently working on to actually have a clinical trial going to replicate all the findings, so the ones with ASD as well as the typically developing. But I wanted to show this as another indicator that we're quite confident this works well. So, which gets me to the next part. What we've done so far, we really focused on other differences in the metabolites. And but these differences in the metabolites, they can come from a variety of sources. I mean, one thing is you may produce too much or too little of a metabolite. And the other thing is the metabolite is used up in reactions. In some cases, these reactions, there may be too much or too little going on. And all of this has an effect on the concentration that you're seeing. Now, all these reactions are affected by cofactors, and most of them are usually enzymes. And so what we want to focus on saying, well, can I develop a model that doesn't just tell me the differences in the metabolites, but are the difference in the reaction rates? And given that can I get the reaction rate really as a parameter that has a population density function? That's what the key is. And see that this population density function is different for children on the autism spectrum versus the typically developing peers. So when we did this, we basically we start out with the concentration balances for the individual components here. Given that we only have one point in time, these tend to be I can't have dynamic balances, so these are just a set of algebraic equations. I have to make a few assumptions, like here between GSH and GSSG, I cannot compute two rate parameters from this. I can compute some net rate or a ratio of one or the other. And there's a few, and so I have to basically simplify this a bit. Similarly, the number of fluxes that I have in here is just too much for the number of measurements with the inputs and outputs. So there have to be some simplifying assumptions. So when we formulate this problem, and we got some data from the literature on what are the removal rates of certain components in there. So we can directly plug this in, and we can then try to solve, we can try to solve this problem, but as I said, there's a few things like you can't get P8 and P9 separately. So we're going to have to basically look at the relationship between the two. And we have to make some other we have to constrain our problems in order to make it solvable. And this was not unexpected. That's given by the structure of the model. And when we do this and then solve for all parameters, you can see that when I look at the parameters from the participants who are typically developing in blue and the ones with ASD in red, you see that there are some parameters where there are big differences. There's some other ones like here where the differences are really minor, next to non-existent. And so then the key is, can we tie what we're seeing here, can we tie this together to either a certain phenomena to explain why are we seeing differences? And secondly, we would be interested in determining then if you wanted to develop some type of drug, you have to figure out which are the parameters that I really want to, or the reactions that I want to up or down regulate. And so what we found here is interesting, and that basically one of the reaction parameters is directly related to oxidative stress. So this kind of ties in the results that we had previously. We also found that another parameter is directly related to the DNA methylation capacity, which basically, this again gives me the link to epigenetics. So even if I look at the difference, the data a different way, the results come out very similar. Now, the last two I was interested in here these MAT1 and MAT3. Now, they are not directly affecting oxidative stress, but they are affected by oxidative stress. So maybe this may be more the question of what's the cause and what the results, but clearly I have essentially three markers that are in some form or another tied to oxidative stress and one to DNA methylation. So this is not surprising. So for this part, what we've shown is that you can actually develop a model, and you find that there are differences in the model you develop for pathways for typically developing individuals and those on the autism spectrum. What's really important here is I wouldn't just want to compute one value and go with that. because the autism spectrum, the spectrum is so wide that having not like a probability density function for the parameters seems a much better, more accurate representation, because the spectrum is broad and the parameters have to somehow reflect that. Just taking the average is not going to tell you the whole story. And interestingly, as I said, oxidative stress and DNA methylation came out to be as the key markers. Again, was saying, well, this is certainly what our study points towards too. Let me summarize overall what we talked about. When you look at metabolites of the folate, 1-carbon metabolism, and the transsulfuration pathways, you find that they have a strong correlation with autism spectrum disorder for our study. They can actually be used as a supportive tool for an ASD diagnosis, and they're also reasonably predictive when it comes to adaptive behavior. And last but not least, the same metabolites, or at least metabolites from the same pathways, taken from a mother during pregnancy, can be used to tell you something about the risk of having a child that will develop ASD. So what is really important here, and I would like to stress this, and this is nice because this is for CAS division. One of the key aspects really was the systems approach. If you did all of this, but you used very simple univariate analysis, as is, quite frankly, is very common in medical research, you are not going to get the results we did. In fact, some of them have been published and they don't come to the same conclusions. But if you reanalyze it using state-of-the-art methods that we commonly use, you actually find that you can get very powerful results. This is what I find very intriguing. So I would like to acknowledge a number of people here. I mean, first of all, my own research group, Daniel Hausmoon did the work for the first part of the talk for the classification, and he's now a postdoc also at the University of Texas at Austin. So, and then Troy Vargason here on the left, he did the work on developing the model where we looked at the parameter distributions. And Katie Hollowood is a student in my group who did the work on the classification for the mothers. I would like to have a number of collaborators, and in particular, I would like to thank Dr. Jill James and Richard Fry, as well as Jim Adams. They're the ones running the trials. I actually do not run clinical trials in my group, but you need to get the data from somewhere, and they're more than happy to supply this. So, and last but not least, the funding agencies that make some of this happen. There's a few references that I've referred to throughout the talk. And last but not least, I'm more than happy to take any questions you might have. Thank you.
Great. Excellent presentation. Thank you for that. We're going to open it up now to questions. You can put a question in the chat window, or you can unmute your microphone and ask a question. So with that, we'll go ahead and start now with any questions that you might have. So I've got one question here. It looks, and you can put the question in the chat window. You can put it privately to me if you'd like me to ask that, or if you can put it to everyone, and then I'll highlight that to Professor Hahn. So let's just go with the first question here. How do so the blood of the mother versus the blood of the fetus, how do the metabolic concentrations relate between the two?
So, excellent question. I'm not sure I have a sufficient answer for you. So when it comes to these particular metabolites, there's no study at all that has ever done this. Yeah, but in general, for other metabolites, people have looked at this. You do find there are differences. Yeah. So for example, which also means that the classifier. I mean, ultimately, the classifier is an equation where I have inputs, and then I'm computing outputs from this. The parameters between the two are very different. Yeah, so I cannot use the same results that I generated for the first part of the talk, where I used the blood of the children, and I'm talking about the mothers. Yeah, and so because people have shown that there are some differences there for other metabolites. Now, for these particular ones, nobody's ever investigated this. So that's because I mean the last thing, getting metabolite, getting blood from the fetus is non-trivial, and it carries a number of risks for other things, and it's just not, to be honest, the risks outweigh the benefit from this.
Another question is, you kind of hinted at this during part 2 of your discussion, that if you have a good classifier and you know the concentrations or combinations of concentrations of the metabolites that predict ASD, the metabolite is an intermediate or product of metabolism, and you kind of hinted at there's some interest for treatment options, where you not only classify, but you could monitor a person's metabolite concentrations over time and evaluate the effectiveness of certain treatments or environmental conditions. Could you say just a little bit more about that? Is that something you're directly involved in, or is that research you're aware of that's external to your group?
Well, it's something that we want to do, and in fact that we're working on it right now. I've seen some very promising results where we can actually can assess the effect of a treatment in the first step on the metabolite concentrations. Yeah, basically, ideally you want the treatment to normalize the metabolite concentration, but more important, you want it to normalize the condition, the ASD severity. We have some data from 3 different treatments that we're assessing in order to see to what degree are they able to do this. It looks promising. The results for the metabolite concentrations look very good, what we have right now. Yeah, but at the same time, we still have to correlate that to how are the individuals feeling afterwards. Yeah, basically the Vineland behavioral score. I have the scores. This is so hot off the press, I haven't even run the correlation yet. On average, they're doing better, but what we're doing is we're looking at person by person. I'm not just interested in the group average. Yeah, and that's something that I may have an answer by the end of the week for you, and hopefully we'll get that published in the next few months. Now, the one thing I should add in this field, there isn't the silver bullet. It's not like you say, oh, you take this and this normalizes everything, we're good. But we can show that certain things, they shift the concentration closer to what you find in the typically developing group. And people, at least on average from what we've seen, tend to do a little better there. But I want to be cautious because I don't want to give people the hope suddenly saying, like, well, Jürgen's working with these, or Jürgen's collaborators are working with these compounds that normalize everything. I wish we were there, but that's a long way off.
Great. Okay, so another question here is, suppose you had unlimited data, how would that change the methods that you use for classification? In particular, things like machine learning, TensorFlow, some of the other methods for artificial intelligence.
So very good questions. We do have, we're in the final stage of putting a paper together where we looked at comparing different approaches, including many of the ones you just mentioned. Yeah. The end result is, so the outcome that we've seen, it does not change. Yeah, so I'm still using the same metabolites, and they're more or less coming in with the same importance as we had. So, but at the same time, if you give me huge amounts of data, I may be looking at deep neural networks, which for my work, we have done this, but there's no benefit for the limited data set that we could find. Yeah, but we're about to publish that because there is a question when people say, well, are your results going to come out dramatically different? And we looked at after second, it's 5, 6, or 7 techniques, but close to half a dozen techniques, and the results come out. They all come out somewhere between 95 and 98% accuracy. So there is not a huge change here or there. Obviously, more data is always better. One thing that I probably would not be doing if I had infinite data, or I would probably not use a brute force approach where we go through all combinations. And I would need some smarter algorithm that hones in on what are the key ones to use rather than us just going through all of them. Yeah, but for the number of inputs we had, this was doable on a desktop PC in a few hours. Yeah, so I'm not too worried about it. But if you said instead of 24 measurements, I'll give you 100, or even 50, I could do this anymore. It doesn't scale that well. We need a different approach then. But these approaches are out there.
One last question here is so that you mentioned a number of environmental conditions, up to 50% environmental versus genetic. What are some of the other factors that you would include in the environmental predictors if you could measure those?
So, yeah, I mean, the key with environmental factors is that basically it's almost anything that comes up. Lifestyle factors are in there. Diet factors are in there. So we're not just talking about how clean is your water and air. Those are factors as well, but it's really environmental factors is anything that affects your body from externally in some form or another. Yeah, and this is where it's really hard to run a study. I mean, people always want the control group. They would like things to be placebo controlled, but when we're talking about including factors from diet and how much sleep you get, and you run studies over months at a time, you can't control these things. It's just not possible for a human being who wants to go out living their life in one form or another, right? And so this is where things get very difficult. But they're all linked to the environmental factors. So it is not just that people think this is just how clean the environment is. That's not at all what it is. It's just any external factors that affect what you get. For example, if you get a number of infections, that would be an environmental factor. So there's lots of things that come into play.
Great. Okay, and looks like we're about out of time right now. We just had one last question here. So when is the blood test going to be available?
I wish I had the answer. Well, look, we have a provisional patent filed, and that's in the last stage of licensing. And we're trying. I mean, the key is you need to replicate the study we did on a much larger cohort. Yeah, and then given that this only is used as a supportive test, this is not going to take 10 years, assuming the funding for the running the trial comes in. That looks very promising right now, and keep in mind, I mean, it's been less than a year that the paper's been published. Almost that doesn't happen that often, but I can't put a date on this. Yeah. Also, you have to understand at some point that if a company wants to pick it up from there, and looks like they might, then this whole thing is on autopilot. So at some point, I may not be the best person to answer that anymore either, but that's okay.
Fantastic. Thank you so much for the presentation and also for answering those questions. Really appreciate the time you've taken today to explain some of your latest research. It looks like some very exciting developments, and we look forward to seeing where this is going to go in the future. So with that, we'll go ahead and wrap up now, and I'll stop the recording. We'll post this online in later today. If you'd like to share it with others, there'll be a link available. Okay, well, thank you, Professor Hahn. I really appreciate taking the time that you took today.
Thank you.